This lecture will go over data as objects and architectures. The readings for this lecture are Wickham, Tidy Data, and Gorgolewski and colleagues, The Brain Imaging Data Structure, a format for organizing and describing outputs of neuroimaging experiments. If you have not read the required readings, please pause, go read those, and come back. In addition, it would be helpful to have the Wickham and Grolman book, R for Data Science. This is a very helpful book for going into depth on many of the topics we're going to be discussing in this lecture. The overall topics for this lecture will be data tables, tidy data, and standardized data architectures. Let's start by talking about data tables. Remember that data science is fundamentally about understanding and gaining an intuition about the systems that produced your data. If we go back to the detective analogy, your data is the evidence that tells a particular story of something that happened. And so, in a fundamental way, you have to have a deep understanding of what data is. We can go from a standard dictionary definition of data as being information, especially facts or numbers, collected to be examined and considered and used to help decision making. This is a pretty good definition of data. Now, data itself can be broken down into two predominant classes or data types. First, there's quantitative data, which is what most people intuitively think of as data. So this is a direct and continuous mapping between variation in an observed phenomenon and a numeric value. Height is a quantitative estimate. The inches you are tall reflects an absolute measure of the environment. So being 5 inches taller than somebody else, those 5-inch metrics match an actual physical distance in height between two individuals. This is contrasted with qualitative data. It's categorical data, essentially, a discrete mapping between a static environmental state or category and an alphanumerical symbol. So classification of group A or group B would be a qualitative or categorical description. Now, this gets to be important later when we start talking about data analysis, but it helps to keep in mind the two general classes of data, continuous and direct mapping versus qualitative and categorical. For today's lecture, we're going to be kind of thinking more about the organization of your data itself. Most of the time, you'll be working with data files. So data files are how your data are stored in a physical medium. They're the computer files that your data is stored in. And there are two types of data files that you're going to be working with. Human readable files. These are files that can be read in text editors by humans. So comma separated files, text files, Word files. They look like this. If you just open them in a text reader, you should be able to see the numbers and the character strings. These are probably the most common type of data you'll work with in most of psychology and a chunk of neuroscience. There are also binary data files that are compressed for storage or security. So if you try to open them with a text editor, you'll see something that looks like this. These are examples like MATLAB files, .r files, which we'll work with a lot if you use SPSS, .save files. The idea behind these binary files is they're usually lots of data, and it would be too big to open in a simple text editor. So you have to compress down the data, or it's encrypted data. Now, the questions you want to ask yourself at the very onset is, what is the information in the file? Do I know how to read the file? And what are my analytical goals? So given a particular data that you've scraped off the internet, pulled from an MRI machine, or pulled from your ePhys machine, or taken from a survey, the first question to ask is, what is my data type? Is it in a human readable format, or is it in a binary format? How do I access the data? That'll determine how you work with it. Now, R, which is the primary analytical tool we'll be using in this class, it allows for you to read either human readable or binary data types. And what it does is it loads it into memory as something called a data table. Data tables are data that is localized into memory and organized in a way that allows for it to be analyzed. So if I visualized it, your data table might look something like this. This is a set of COVID hospitalization numbers that I pulled from the COVID tracking project. The idea of data tables is that it is your data organized into system memory. So it's usually represented as a two-dimensional array, like what you're seeing here. And data tables are actually what most people in our field think of when they talk about their data, in quotes. So data tables themselves are the primary data objects that you'll be working with in your tutorials in this course and in your research. Most of the time, data tables and their form are overlooked. The goal of this lecture is to show you today how you need to take particular care in thinking about your data tables, because how they are organized will determine the efficiency of your analysis. And this brings us to the topic of tidy data. What is tidy data? Tidy data, which was proposed in the Wickham article, is a standard way of mapping the meaning of a dataset to its structure. Fundamentally, it's a way of having a logic to organizing data tables in a way that preserves the meaning of the data itself. And the basic properties of tidy data are that each variable forms a single column. Every column should be an independent variable. Each observation forms a row. So in this case, the date of COVID deaths and COVID hospitalizations is a separate row. And most importantly, each observational unit forms a table. An observational unit, in this case, is a target of a question. So if I'm asking about properties of variation in COVID deaths, hospitalizations, and diagnoses, that would be a single observational unit. We'll get into a little bit more detail about this in a little bit. Before we dive too deep into Wickham's tidy data, it helps to go over and confirm some key terms or key definitions. First, a data set we'll define as a collection of values. Really just any collection of value. What's a value? A value is an analytical unit. So it's a number or a string. A variable is a set of values that measure the same attribute across units. In the COVID example, it's daily hospitalization numbers across observational units, which were, in that case, dates. Observation, all values measured on the same unit. And a table is a collection of variables and observations organized in a two-dimensional array. These are the fundamental terms as we will use them in the discussion of tidy data, and indeed in the other lectures of this class as well. In order to understand tidy data, we're going to use the contrast to what I'm going to call dirty data tables. And I'm just using the concept word dirty in the contrastive to the word tidy. It doesn't actually mean there's anything wrong or bad, although we'll see some anomalies in these tables. Mostly it's just used to contrast with the concept of a tidy table, which conforms to Wickham's definition of tidy data. So for example, looking at this data table, which is just a toy data table, where we have person, a treatment condition, A or B, and some outcome result. This would qualify as a dirty table. And let me show you why. Where are the values? Well, that's the individual items in each cell. The variables are treatment, person, result. And the observations are the outcome for each person in each treatment. Now you notice that here the observational unit is person, and person is repeated twice across separate rows. So each person is not an individual row. What you have is Joe is represented in the first row and the fourth row, June in the second and the fifth, Mary in the third and the sixth. So this violates that concept of each observational unit, a person, being a single row. So in order to tidy the data table, you would want to have it look something like this, with the missing measurement in Joe for condition A, the response in condition B. Now, each person, observational unit, is a row and each variable is a column. So each variable A and B forms a column as well as person. Each observation forms a row, and each observational unit forms a table, the single table of the treatment condition effects for these three participants. Now, tidy data is the ideal data form, but the goal of understanding how to get to tidy requires understanding typical problems that you see in data tables. So next we're going to go into problems that lead to dirty data tables and solutions for how to clean them and make them tidy. The first problem we're going to go over is the problem where column headers are values, not variable names. And the solution we're going to go over is the concept of melting. Let's see this by example. Here is a dirty data table. So here you can see in this representation, each column represents a value of a condition. So you have A, B, and C. Now here, what we're going to do, in contrast to the example that I used at the beginning, is we want to have condition be the main observational unit, not individual. So in order to fix this, what we're going to do is we're going to melt the data, which is unify data across columns that are subordinate to a common variable. So the common variable here that we really want to get transferred to a column is the variable of condition. So now we have condition. So each row now represents a unique observation across conditions. Notice here how in the prior example that I used at the beginning, this is almost an inverted version of the form. In the prior case where we had Joe and Mary and the conditions, we wanted the single observational unit to be person. Here, the analytical goal is condition, and the label XYZ is a repeat measure. So you can start to see how the concept of a tidy data table depends a lot on your analytical goals. Another problem that comes up is multiple variables are stored in one column. And the solution to this is something known as splitting. So let's see this by example again. Here is a dirty data table. So this is the case where we have country as a variable, group as a variable, and cases as a variable. Now notice that in the group column, we actually have two variables. We have gender and we have age group, which in this case are binned between 0 and 14, 15 to 24. So what we want to do is we want to split down the middle of that variable and separate it into two variables. We want to take that one column and make it two. Splitting is separating one column with multiple variables into multiple columns with one variable. So the tidy data variant of this looks like this. So now all we've done is taken that group variable and turned it into a gender variable and an age group variable. Another problem we run into are that variables are stored in both rows and columns. And the solution to this is something known as casting. Here you'll notice that we have a column for date, a column for measure, and a column for value. So here the column for measure actually identifies two separate variables, a variable for the mean and a variable for the variance. So what we want to do is cast those two separate measures. Now notice, unlike the last one, they're not consolidated within a cell, but they happen across rows. We have mean and variance for the same date on two separate rows. We want to recast those rows into separate variables themselves. So the solution to tidying this data table, where we have this repeat of date observations, depending on whether you're talking about a mean or a variance, is to just make them their own columnar variables. So now we have each date is in a unique row, and each column is either the mean or the variance. So now they're separate data columns. Another problem we run into with data tables is when you have multiple types of observations that are stored in the same table. And the solution to this is something known as parsing. So let's consider this example. I have a data table of document ID numbers, DOIs, for different papers. Let's say one paper is authored by Versteinen with the title Data Rules. It was published in 2017. We want to track the citation rate. So we have a column for citations, years, the title of the paper, the author, the first author, and the DOI number. There's a second paper with DOI number 002, published by Holt, and then with the title Theory Rules, published in 2017, and we have its relevant citations. Now, here, the types of observations that we have in this single data table are data about paper identity and data about citations across time. And what we want to do is we want to parse those or disentangle those into separate data tables. Essentially, what we want to do is separate out the citations across time information from the information about the data table itself, or about the papers themselves. Now, with data tables, the nice thing about this is that if you parse them, you can still connect them. So if we parse these by taking one table with multiple observational units and separate it into multiple tables with unique observational units, we can have a table that defines article identity, and we can have a table that describes the citations by year, linking it with a single ID number, right? So the DOI exists across both tables, and we can link these tables together. So I now have collapsed the observational units to separate data tables, but I have information that allows for me to use them together in order to identify what's the relevant information that I need to know about the particular paper number and how it tracks across time. That's the key to parsing. Finally, a major problem that you see in data tables is single observational units stored in multiple tables. And this is often occurs when you're doing your own data collection on subjects, single subjects, let's say. And you need to put them together into a broad experimental data table. And the way to do this is just concatenation. So let's say you're running a cognitive psychology experiment. You have conditions A, B, and A. And you're testing reaction times, and so you collect a data table for subject 1, you run another subject the next day, you get a separate data table, and all you want to do is you want to look at these responses of conditions by measuring variability across conditions across subjects. So you want to concatenate or merge multiple data tables with the same observational units into a single data table. So really you're just concatenating these two together. So you have subject ID, trial, condition, and RT are their consistent tidy format. So we have observational units are trial and condition responses, and we have subject as the repeated measure here. So these 5 problems and these 5 solutions represent the major kind of mechanisms of making untidy data tidy. Melting, splitting, casting, parsing, and concatenation all are tools that you have for making individual data tables tidy. The important part to really bear in mind is that how you set up your tidy data table depends a lot on your analytic goals. So you want to have a logic for thinking about the specific way you're going to be looking at your data when thinking about how to make the relevant data tables tidy. Often this is overlooked in research, particularly if you inherit data tables, let's say working with already existing data collected in another lab or publicly available data that's often in messy forms. So you want to get comfortable with the tools for making tidy data tables in order to really clean up the architecture of the tables themselves that you're going to be using as your primary point of data analysis. Along with organizing the data tables, you want to think about having a standardized data architecture for the files themselves. So we've spent a lot of time talking about the tables that you're going to be using for running your individual analyses and visualizations, but you also want to have a systematic way of organizing the files themselves, including pointers to code that you use to analyze and your data files themselves. And I'll explain why that is in a second. So moving beyond data tables, having a data file standardization or standardization of the physical forms of your data basically boils down to having a systematic file formats, directory organization, and naming logic that allows for effective collaboration and sharing. The point of having a standardized data architecture is more about sharing and collaborating than it is about setting up a pipeline for yourself. And there are many advantages to developing a core logic of data standardization for your data files and data directories. So first, and this is pointed out in the Gorgolewski paper, having a standardized data architecture minimizes curation demands. It makes it possible for those not involved in the research to understand the data with minimal instructions. If you have a common logic and people know that logic, there is not a lot of effort needed on your part as sharing the data to communicate how the data is organized. People will understand it because it relies on that common logic. It reduces the errors in your data sets as well. Having a common standardized data architecture reduces errors attributed to misunderstanding the meaning of a given datum. It optimizes data use by facilitating aggregation across studies to promote reuse and reanalyses. If you have a community of people who are using data organized in the same architectural logic, it facilitates an entire group of people coalescing together and sharing data, and then more importantly, aggregating data of the same type. This is very common in the field of, or becoming more common in the field of fMRI, using the BIDS data architecture, which was the Gorgolewski paper, so people can bundle together data from different studies because they're all organized in the same way. They're all in the same format. And finally, it promotes or fosters automation. So the idea that you want to foster the development of tools that work across data sets. If you have your data organized in a common logic and you build a tool that relies on that logic, then anybody can take that tool and apply it to their data, which also has the same architecture. There is a very clean beauty in that, because now you don't have to really take time to understand how people organize their data or where the metadata is or what the file naming convention is, what a subject is. As if there's a standardized data architecture and you want to write a tool to automate some process, that tool will work on any data that follows that same common architecture. And this is the main argument that Gorgolewski and colleagues put forward when they proposed a common data architecture for functional brain imaging that, as I will point out, actually works for really any type of data that we're going to be working in neuroscience or psychology. For formal data architectures, you want to understand first, what are they before we kind of dig into how they're organized. So a data architecture itself is just a formal logic of data and directory organization. How do you label your files? How do you label your directories? Where do you store the metadata? Where do you put the meaningful information that describes, let's say, the organization of the experiment? Where do you put the meaningful metadata that describes what a trial is or how a subject was run? And this was the major inspiration for BID. So this is the image I pulled from the Gorgolewski paper. On the left, you see the standard raw architecture of neuroimaging files as they come off the scanner. On the right, you see the BID's standardized data architecture. So notice first the hierarchy is standardized. You first have some name for the data set, and then underneath that, you'll have directories for the subject ID. In each subject ID, you'll have subset directories for the anatomy, functional imaging, diffusion imaging. In each directory, you have a common file type. What you're seeing here is like the .nii.gz file type for the main data. Also .json files, which are metadata files. There's also a common naming convention. You see sub-01 underscore T1W.nii. That tells me right away that that's the T1 anatomical image for subject 1. If you go into the functional images, sub-01 underscore task.rest tells me that this is the resting state bold image. And the JSON file probably tells me meaningful information about how that was collected. Because I understand the logic of the BID's organization, I didn't need somebody to tell me where the resting state data was, where the diffusion imaging data was, where the data vectors were for, or the Q sampling vectors were for the diffusion imaging. All of that's there. There's a participant text file at the top that tells me everything I need to know about the participants in this experiment. Somebody could zip this data set, this whole directory, pass it to me, and I will understand, at least from a data logic perspective, everything I need to know about analyzing this data. Now this will work with just behavioral data. You could just have a behavioral directory and follow the same logic. This will work with EEG data. The idea of BIDs is to make a universal standard data architecture that works across neuroscience, and I'm going to argue will also work for psychology. In fact, in my lab, all of the output of our behavioral experiments must be BIDs compliant as well. So the nice thing about this is now I can pass this along to anybody who wants to use the data, and I can share it, and they can use it right away without having to contact my lab, without having to have any possible misunderstandings about which data type is which. In order to do this, though, you have to pay attention to certain things. File types. There must be a common community agreement, or at least agreement within your lab or your research team as to what your file types are. So for example, you see the .nii.gz format. That's a nifty file format. There are other types of file formats for neuroimaging data. There's bricks, there's DICOMs. You have to agree what your file types are and what you want them to set as. Also the JSON file, the .json file extension, that's a standard metadata type, but there are other ways of storing metadata. So you have to have some agreement on what the file types are. There must be some naming logic to the directories and the file names. There must be some logic to the directory hierarchy. Here you see it goes from experiment to subject to data type. If you have a set of logic of how you organize your directories, that's fine, but you just want to keep it consistent. You want to think about your documentation and your metadata. How do you want to store your metadata? Where does the metadata go? How does it link to the files? You don't have to use the BIDs format. I'm going to argue that if you're doing research in psychology and neuroscience, you're going to want to adopt the BIDs format because it's already done for you. But if not, I highly recommend defining a data architecture and a standardized data architecture for your lab or your research group that you can easily communicate to others so that they can learn that logic and you can give them data and they can follow it. So I hope that this lecture has made clear that having standardization, having ways of cleaning and organizing your data tables, as well as your data files and directory structures themselves, is incredibly useful for a host of reasons. For sharing data, for organizing your data. So the take-home message should be that having a standard logic for your data tables and files reduces the many sources of errors that happen in research and facilitates data and code sharing. It goes towards that value of openness, which we talked about in the value of openness lecture. dig into the tutorials on tidy data and data architectures. Read up on standard data architectures in your field. Understand how to make optimal data objects, because if you could do those, working and developing automated pipelines for producing your results and allowing people to replicate results naturally fall out of that. So this is one of those things we often overlook in data science, but I think is important. You want to think about how your data is organized and stored in order to make your research more systematic and more shareable.